Hello dear students, today we are going to discuss some important concepts regarding complement system like what is complement, why is it important and how complement system was discovered. Secondly, we will try to know the proteins of the complement pathway and the related nomenclature. And finally, we will try to understand the regulation of complement activity. So students, coming first to the concept of complement and what is its importance. Blood contains a group of serum proteins that can act in conjunction with the cells of the immune system to eliminate foreign cells. These proteins are called as complement proteins. A large number of complement proteins can occur in an inactive form as zymogens. These zymogens are activated by proteolytic cleavage, which removes the inflammatory fragment and exposes the active site. Once activated, the complement system can bring about a variety of responses. For example, it can initiate an inflammatory response or cause the destruction of the bacteria, parasites, viruses, infected cells or red blood cells. In addition, it can facilitate the clearance of the dead cells and the immune complexes. Thus, the complement system is the major effector of the humoral branch of immune system. Now, after understanding what complement is, let us have a look at its history. Research on the complement began in the 1890s when the Jules Board at the Pasteur Institute in Paris showed that sheep antiserum to the bacteria Vibrio cholera causes the lysis of the bacteria and that heating the antisera destroys its bacteriolytic activity. Surprisingly, the ability to lyse the bacteria was restored to the heated serum by adding a fresh serum that contained no antibodies directed against the bacterium and was unable to kill that bacteria by itself. Board correctly reasoned that the bacteriolytic activity requires two different substances. First, the specific antibacterial antibodies which survive the heating process, and the second, heat sensitive component responsible for the lytic activity. Board devised a simple test for the lytic activity, the easily dissected lysis of the antibody coated red blood cells called hemolysis. The discovery of the complement is usually attributed to the Board. Paul Ehrlich in Berlin independently carried out similar experiment and coined the term complement, defining it as the activity of the blood serum that completes the action of the antibody. Pillemer and his colleagues in 1941 described the partial fractionation of C1 from the mixture of the proteins contained in the C1. In 1960, Mueller purified major plasma protein, a C1 component which is present at the concentration of 1 gram per liter. Mueller also demonstrated that it corresponded to C3. Scott Nelson and his colleagues were able to purify the 9 active components now recognized to participate in the sequence of hemolysis by the complement classical pathway. An antibody-independent pathway of the complement activation was first suggested by Peterman and his colleagues in the early 1950s, who identified a substance which they called propertin, which was involved in the activation of the complement pathway by yeast and certain bacteria. Now, dear students, we will try to know what are the complement components. The complement system is composed of several unique plasma proteins. These proteins are synthesized mainly by the liver hepatocytes. Although a significant amount are also synthesized by the blood monocytes, tissue macrophages, epithelial cells of the gastrointestinal tract and the urinogenital tract. These proteins mostly circulate in the blood in an inactive form as zymogens. These zymogens are activated by the proteolytic cleavage, which removes the inhibitory fragment and exposes the active site. The complement reaction sequence starts with an active enzymatic cascade. After knowing the complement components, let us try to understand the nomenclature. Complement components are designated by numerals C1 to C9, by letter symbols, for example, factor D, or by trivial names, for example, homologous restriction factor. Peptide fragments formed by activation of a complement are denoted by the small letters. In most cases, the small fragment resulting from a cleavage of a component is designated by the A and the larger fragment is designated by B. But in case of the C2, C2 is an exception. C2A is a larger fragment, cleavage fragment, because unfortunately the small fragment of the C2 was originally designated as C2B. However, following a uniform nomenclature is advocated and is being presently followed. The larger fragments bind to the target near the site of activation and cause the cell lysis, and the smaller fragments diffuses away from the site and can initiate the localized inflammatory responses by binding to the specific receptors. The complement fragments interact with one another to form functional complexes. These complexes that have enzymatic activity are designated by a bar or a number or a symbol. The proteins of the alternative pathway are assigned by the letters preceded by the symbol F. Sometimes the F is omitted, so factor B may be represented simply by writing B. Now, complement functions. After initial activation, the various complement components interact in a highly regulated cascade to carry out a number of basic functions, including the lysis of the cells, bacteria, and viruses, opsonization, which promotes phagocytosis of a particular antigens binding to a specific complement receptors on the cells of the immune system, triggering the specific cell function, inflammation, and secretion of the immunoregulatory molecules. Immune clearance, which removes the immune complexes from the circulation and deposits them in the spleen and liver. Finally, after knowing the complement components, we need to know how they are activated. So, dear students, coming to the complement activation. There are three converging pathways of complement activation. Each one is stimulated by a specific set of stimuli. All the three pathways converge to form a one common thing, a membrane attack complex that forms the pore in the membrane of the pathogen, causing the lysis and eventually death. These pathways are first the classical pathway, then second is the alternative pathway, and the third is the mannan binding lectin pathway. So students, coming first to the classical pathway. The classical pathway of a complement system consists of blood proteins that mediate the specific antibody response designated as C1, C2, C3, C4, C5, C6, C7, C8 and C9. C1 in the serum is a macromolecular complex consisting of C1q and two molecules of each, C1R and C1S, held together in a complex stabilized by the calcium ions. C1q molecule is composed of 18 polypeptide chains that associate to form 6 collision-like triple helical arms, the tips of which bind to the exposure C1q, the binding sites in the CH domain of the antibody molecule. Each C1R and C1S monomer contains a catalytic domain and an interactive domain. The latter facilitates the interaction with C1q or with each other. C2 is a proenzyme which acts as a substrate for C1S, which cleaves C2 into C2A and C2B. Then C2B with C4B enzymatically activates C3. C3, the major constituent of the complement system, is present in the plasma at a concentration of approximately 1 gram per liter. C3 contains two polypeptide chains, alpha and beta. C3 belongs to a family of proteins containing an unusual post-transcriptional modification to their structure. This is an internal thioester bond consisting of a glutamine residue whose carboxyl group is linked to the sulfidyl group of a near cysteine residue. This bond is metastable and the electrophilic carboxyl group of the glutamine is susceptible to the attack by the adjacent nucleophilic groups such as hydroxyl group and amine group in the adjacent proteins and carbohydrates. This reaction allows the glutamine to bind covalently to other molecules and is the mechanism by which C3 can become linked to them. C4. The C4 component is a glycoprotein containing 3 polypeptide chains, alpha, beta and gamma. The C4 also contains an internal thioester bond within a sequence that is closely homologous to the one in C3. There are 2 isotopes of C4, C4A and C4B. C4A binds to amine groups forming an amide bond, whereas C4B mainly binds to the hydroxyl group producing an ester linkage. The C4A binds mainly to the amine group within the proteins and C4B to the hydroxyl group within carbohydrates. C5. This component of the complement is activated by a trimolecular complex C4B2A3B called C5 convertase. The C3b component of this complex binds C5 and alters its conformation so that C4b2a complex can cleave C5 into C5a, which diffuses away, and C5b, which attaches to the C6 and initiate the formation of the membrane attack complex in a sequence. Rest of the proteins of the complement C6 to C9 forms an integral part of the MAC. Thus, the activation of the classical pathway begins with the formation of the antigen-antibody complex or binding of an antibody on a suitable large antigen. IgM and certain classes of IgG are known to activate the classical complement pathway. The C1 is a large multimeric plasma protein complex. The catalytic action of C1 is initiated by C1R, tetramer that is non-covalently associated with the C1q subunit. C1s and C1R are proteolytic enzymes that remain inactive till the C1 molecule binds to the immune complex. The binding of the heads of the C1q to the IgM or IgG molecule induce a conformational change that leads to an enzymatic activation of the associated C1R to activate. The activated protease then cleaves C1s to a similar active enzyme C1s. The C1s has 2 substrates of the classical complement pathway, C4 and C2. The C4 component is the next complement component that is activated and then cleaved by C1s to a smaller fragment C4a and a large fragment C4b molecule. The smaller one diffuses away and functions as a mediator of the inflammation. Then C4b molecule forms covalent bond with the hydroxyl or amine group of the nearby target surface in the vicinity of C1. The next plasma protein to be activated is the C2. It is cleaved by C1s into a larger fragment C2a and a smaller one C2b. C2b then diffuses away and C2a remains physically associated with the C4b on the cell surface. The resulting C4b2a complex is called as C3 convertase, having the ability to cleave the C3. Henceforth, serum component C3 is then cleaved by C3 convertase into C3a and C3b. C3a is removed away and C3b attaches to the cell surface molecule near C4b2a to form a new complex C4b2a3b, which functions as C5 convertase. This C5 convertase has a catalytic activity. This C5 convertase catalyzes the enzymatic cleavage of C5 into C5a and C5b. C6 and C7 rapidly binds to the C5b, forming C5b67 unstable complex, and once it becomes attached to the membrane, it becomes stable. The addition of the C8 and C9 results in the formation of a complex C5b6789, which is called as membrane attack complex or MAC. This complex creates the pore in the target membrane. and helps in the lysis of the cell. Now dear students, coming to alternate pathway. This pathway was alternative of the complement activation and could be activated by yeast or even by various bacterial polysaccharides. This complement activation can occur in absence of the antibodies and hence can be considered as the component of the innate immunity. The alternative pathway involves 4 serum proteins: C3, factor B and D and propodin. The alternative pathway components are called as factors and are designated by capital letter symbols. In contrast with the classical pathway, it does not depend on antibody for its activation. The alternative pathway is activated by the spontaneous hydrolysis of C3 so as to generate large amount of C3b and C3a. Then this C3b binds to a suitable surface, for example, outer membrane lipopolysaccharide of bacteria. Once bound to the surface, it attracts another factor, plasma protein B. This binding of B by C3b allows another alternative complement protein D to cleave B into BA and BB. BB then remains associated with C3b to form C3b capital B small b complex. This complex is highly unstable and gets stabilized by another alternative pathway member called propodin or protein P, which prevents spontaneous decay of BB from enzyme. This relatively stable C3BBB complex is a potent C3 convertase, capable of cleaving C3 into C3A and C3B. Some of the C3b molecules generated by C3 convertase bind to C3 convertase itself to form new complex C3b BB3B. This is referred to as C5 convertase, which functions to cleave C5 into C5A and C5B. This C5b component binds to the microbial surface and initiates the formation of membrane attack complex. Now coming to the third pathway, that is the mannan binding lectin pathway. This pathway, which has been recently discovered, is an additional pathway by which complement system can be activated. Mannan binding lectin is an acute phase protein. It is present in low concentration in the plasma of an individual and its concentration is increased in inflammatory responses. MBL has C1q-like structure and is 6-headed. It specifically binds to the mannose residues present on the surface of a microorganism. After it is binding to the pathogen, 2 MBL associated serine protease zymogens or MASP bind to it. MASP1 and MASP2 share homology with C1r and C1s respectively and mimic their activity. When the MBL complexes with the MASP and this MASP MBL complex binds to the pathogen surface, MASP1 and MASP2 are activated to cleave C4 and C2 to form C4b2A. This complex, which is classical C3 convertase, gets deposited on the surface of the microorganism. Thus, lectin pathway activates the complement activation almost the same way as classical pathway. It is considered as another important arm of the innate defense mechanism, as C3 convertase is formed without the need for specific antigen-antibody complex. So, my dear students, in conclusion, we may say that complement is an important component of innate immune system that is crucial for defense from microbial infections and for clearance of immune complexes and injured cells. Complement system consists of about 30 serum proteins that are mostly in an inactive form called as zymogens. These inactive proteins become activated when they are cleaved into an active fragment. This activation occurs through three different pathways: classical pathway, alternate pathway, and the lectin pathway, or the mannan binding lectin pathway. All these three pathways lead to the production of C3 convertase. Once this happens, all the three pathways behave in an identical manner. That is, they cleave C3 into C3a and C3b. C3b binds to the another complement component to form C5 convertase. Then this cleaves C5 into C5a and C5b, which later combines with the other proteins like C6, C7, C8, C9 to form membrane attack complex. This MAC finally forms the pores in the membrane of the pathogen, causing its lysis and eventually death. So, I hope you have enjoyed the lecture.